Bismillahirrahmanirrahim. Welcome back to AmreenaMehrajTutorials. My dear students, today we'll continue understanding about mutagens in part 2. I hope you have gone through part 1 and you have find that interesting. My dear students, in part 2 we'll be understanding about the chemical mutagens in detail. So let's get started and try and understand about chemical mutagens in detail in today's video, that's mutagens part 2. Make sure you have watched part 1. Don't forget to like, share and subscribe to my channel, and meanwhile don't forget to comment down in the comment section if you have any suggestions for me. Thank you so much. Let's get started.
Anything. Time doesn't wait for anybody. Okay? No, technology is over there. It's been getting translated. That's why I speak in English, because I want it to be usable. You can turn on your, captions, and everything will appear automatically on your screen. So number one, mutagen, under which category? Chemical mutagens. Okay.
My dear students, if you're watching part 2 and you haven't watched part 1 for mutagens, no worries at all. You will watch part 1 later on. But before moving ahead, let you have an idea about the classification of chemical mutagens first. I'll be showing you a short clip of 3 minutes. Pay attention.
So chemical mutagens. Let me give you the first of all, let me give you classification of chemical mutagens. So I'll be now over here. We have to discuss about chemicals which cause mutations. So they were physical agents, huh? You got this idea. Here we talk about what? We talk about radiations and we talk about temperature. Both of them are physical agents. Now we have to talk about chemicals. So out of chemicals, my dear students, number one, I will be teaching you about, inshallah, alkylating agents. From chemistry, you might know. Alkylating agents, like I'll be talking about dimethylsulphate. Example, this is one of the chemical. Okay. Similarly, I will be talking about, I have written bigger, okay. I already have less space. Second chemical which I'll be talking about, I'll write down properly now. Second is base analogues. Base analogues. Over here, let me give you an example. It's 5-bromouracil, a chemical, which is a base analog. What it means, I will tell you right now, in a while. Okay, so number one are alkylating agents. Number two, chemicals are called base analogues. Number third chemical is called deaminating agent. You already know this, huh? Deaminating agents. They will cause deamination. One of the best example of deaminating agent is nitrous acid, HNO2. The fourth one I'll be talking about today is acridines. Basically they are the dyes. Over here I'll be giving you an example of, maybe we can say simply acridine orange. It's a dye basically. It's also a chemical, ORANGE, spelling is incorrect. Orange. Acridine orange is the example. So we are going to talk about how many? This is third, this is fourth. We are going to talk about how many? Yeah, we are going to talk about alkylating agents, the chemical which will cause mutation, base analogues, chemical which will cause mutation, deaminating agents, chemical which will cause mutation, and acridines, the chemical which will cause mutation. Over here, remember the example. One example for alkylating agent is DMS. Example is 5-bromouracil. Example is nitrous acid. Example is acridine orange. Okay, clear to you people? Now let we take this category followed by this, followed by this, followed by this. Then it will be our lecture done. I hope you are finding it interesting. I hope I'm trying to make it as interesting as possible. So keep on understanding, keep on learning, keep that urge in your mind, because time is flying like anything. Time doesn't wait for anybody. That's why I speak in English, so that you'll be benefited more. Number one is chemical mutagens. So chemical mutagens we are basically discussing, and number one out of them will be, let us say for example take alkylating agents. Okay, let's take alkylating agents. So I have this opinion, I have this concept in my mind that you might be knowing about these things because you're class 12 students. So alkylating agents are what? They'll be doing what? Simply, they'll be doing alkylation. They'll be doing alkylation. What is that alkylation? It's addition of what? It could be like addition of methyl group. It could be addition of ethyl group. They are all alkyl groups. So what are you doing? Basically, there will be some chemical which will be capable of adding alkyl group. So what is this group basically? This is which group? This is alkyl group. They'll be added. Okay, either methyl group will be added or ethyl group will be added. Now, ma'am, where it will be added? In the DNA. It will be added to the nitrogenous bases. So, alkylation will happen to the nitrogenous bases. So, we are not bothered about the phosphate backbone and the sugar. These things, these damages happen to the nitrogenous bases. Now, which is the chemical which can do this alkylation? Very important. The examples of alkylating agents. The examples of alkylating agents which can cause alkylation. They include DMS. DMS is dimethylsulphate. So it's a reagent, a chemical which can, basically it can donate methyl group. So if you have some reactant, it will react with that and it will convert that compound into what? Methylated compound. So it is capable of performing what? Methylation. So dimethylsulphate is one example of alkylating agents. Similarly, you have, okay, I want to place it somewhere where from I will get it easily. Okay, dimethyl sulfate is one example. Another example, these examples are very important. Another is dimethyl nitrosamine. This is also an alkylating agent. See, it can also donate methyl group. Number third is, my dear students, nitrogen mustard. EMS is ethyl. Okay, methyl sulfonate. Okay, ethyl methyl sulfonate. Yeah, you just have to remember the names. Okay, another I will give you is S-adenosylmethionine. S-adenosylmethionine. These are some of the examples of your what? Alkylating agents. They can cause alkylation. Now, my dear students, let me tell you what they can do basically is they can add what? Either they will add what? They will cause addition of, first of all, let me write down. Addition of alkyl group to nitrogenous bases. Okay, now either what will be added, my dear students, either you have addition of methyl group, the CH3 group. Accordingly, we will call this alkylation what? You will call it methylation of nitrogenous base. Either you have addition of what? Usually you will find these two groups. Addition of ethyl group, accordingly you will call it ethylation. Okay, so you have CH3 being added, accordingly methylation. Okay, now my dear students, let me give you one example. Say for example you have guanine. Okay, normally you have guanine. Once this guanine will be exposed, or it will react, or you will add say for example EMS, ethyl methyl sulfonate. Where is it? This. You added this compound into the cell. Okay, what will this cause? This will convert this guanine into methyl guanine, and specifically it will target the 6th oxygen of the group. So at oxygen 6th position, where you will find the oxygen, that will be methylated and you will get O6 methyl guanine. What is it? Simply, this is simply methylated guanine. What is it? Methylated guanine. Now this will not behave normally. See, guanine normally it base pairs with what? It was supposed to base pair with what? Guanine base pairs with what? Come on, answer. This base pairs with what? G base pairs with cytosine. But this methylated guanine will not base pair with cytosine. It cannot, base pair with cytosine. If it cannot base pair with cytosine, tell me, my dear students, won't this affect the pairing of nitrogenous bases in DNA? Won't the pairing of the nitrogenous bases in DNA be affected because the property got affected? When the pairing of the nitrogenous bases is affected, there are more chances of substitution type of mutation. I hope you know that. There are more chances of substitution type of mutation where one base is, changed with some other base. So consequently, methylation resulted in mutation, substitution type of mutation. This is about what? This was about alkylating agents. So what do you need to remember is alkylating agents add alkyl groups, be it methyl group, ethyl group. I give you example where guanine is methylated into methyl guanine. Now if it's converted into methyl guanine, its pairing properties will not remain normal. Now it cannot base pair with its normal pairing partner, that is cytosine. It will not base pair. If it will not base pair, there will be definitely difference in the normal structure of the DNA. Now if this defective DNA will undergo replication, there are more chances that at this particular place where you have methyl guanine, at that place the correct base will not be added to the newly synthesized DNA strand. So DNA polymerase will mistake this and it will substitute a wrong base in the new DNA. Okay, this was about alkylating agents. Now we need to go to which, let us say for example, let us take the deaminating agents. Let's take deaminating agents. Okay, so can you recollect from your notebook, if you are copying along with me, can you recollect one example of deaminating agent I have given you earlier? Just 5 minutes before I gave you, I think less than 5 minutes. It didn't took me 5 minutes to complete this. Okay. The second one is, I said I'll take, second one is, I give you a word, deaminating agents. Can you quote any example? Yeah, deaminating agent. What will they do? They will cause, let me write down because you then ask. They will cause deamination. That means what, my dear students? Removal of NH3 group. It's basically better to write down a removal of removal of amino group. Okay, deamination. They will cause deamination. Again of what? Deamination of nitrogenous bases. Now, I have given you earlier this example of a deaminating agent. I told you that the versatile deaminating agent is what? What is this called? Excellent. It's called nitrous acid. Remember one thing, where from nitrous acid will be produced. Who will be the precursor for nitrous acid? You will have sodium nitrate, or you will have sodium nitrite, or you will have nitrosamines. Okay, these are what? These three are what? They are the precursors. Precursors of what? Precursors of nitrous acid. So nitrous acid will be produced from these three substances. Okay. Now if you have nitrous acid in a cell, it will again target what? It will target the nitrogenous bases. And what is its job? Its job is to take out amino group. Okay. It will take out the amino group, thereby causing deamination. Say for example, it will attack, I have told you in spontaneous mutation, it will attack cytosine. Okay, cytosine has amino group that will be removed. So what is it? Removal of NH3. What is it? Removal of amino group. Now once cytosine, you will take amino group out of cytosine. What is the product? So what happened? Cytosine got converted into uracil. Who is the causative agent? Who is the mutagenic agent? Who is the mutagen over here? HNO2 is performing this reaction. So this is the reaction catalyzing substance. HNO2 is performing this step. HNO2 is responsible for taking away this amino group. Now similarly, I told you earlier, once we started the lecture, similarly adenine will also undergo deamination, and it will be converted into what? Adenine will be converted into hypoxanthine. Okay. Similarly, you have, my dear students, what? Guanine. Yeah. Guanine will be converted into xanthine. Okay. So what are you seeing? what is being removed? Amino group removed, amino group removed, so NH3 removed, NH3 removed. So what is happening? What is happening basically is the deamination, and property is changing. You get uracil, hypoxanthine, xanthine. Okay. Now, my dear students, cytosine normally base pairs with what? Cytosine is supposed to base pair with guanine. Okay. Cytosine base pairs with what? Let me show you. Cytosine base pairs with what? It base pairs with guanine. But uracil will not base pair with guanine. It will base pair with adenine. Okay, adenine usually no, not a correct statement. Adenine base pairs with what? It pairs with thymine, but hypoxanthine will base pair with cytosine. See, the characteristics are changing. Guanine pairs with cytosine, but this xanthine doesn't pair with cytosine. It pairs with thymine. See what has happened? A wrong base attached, a wrong base attached, a wrong base attached. What will happen? What do you call this? Substitution type of mutation. See, once you have this thing happening in the parental DNA, okay, so in parental DNA there was cytosine, it got converted into uracil, this got converted into hypoxanthine, this got converted into xanthine. Now you have these bases in parental DNA. Once replication will start taking place, this will bind, this will attach adenine in nilly strand is synthesized DNA strand, because this acts as a template. Now template is incorrect. Incorrect bases will be added in the DNA. So substitution will happen. Substitution of wrong bases will happen in the DNA. That's why these are causing mutation. But who is the causative agent? Causative agent, the reason is the deaminating agent. Earlier the reason was alkylating agent. Earlier problem was caused by EMS. Now here problem is caused by nitrous acid. Okay, this is how deamination is happening in your cells. So deamination is a simple process of the removal of the amine group, and who is causing this deamination is nitrous acid. Now the third type of chemical mutagen is, let us say for example take base analogues. Okay, let us take base analogues. Base analogues are very interesting yet again. You will find out the details about the same things in higher classes, but I am trying to give you as standard information as possible. But I don't want to overburdenize you people, because I don't want to give you that information which you don't need. So what is the example over here? Which is the agent we saw over here? Nitrous acid. So this over here we are trying to give the example so that we can revise at the end. The third chemical mutagen, let us say for example take base analogues. They are very simple. Base analogues. Base analogues. I hope you are able to understand my handwriting. So base analogues, my dear student, as the name indicates, analogues. There will be some chemical which will be resembling a base, but that won't be the correct base. But it's an analog. It resembles. Okay. So what are base analogues? Basically, I need to write down over here. That's very important. Base analogues will be, such chemicals which will what? This stands for which? Which will, which resemble with nitrogenous bases. So I'm writing like this, nitrogenous bases. They resemble with nitrogenous bases, and once this, and let me write down, and get what? They get incorporated into DNA during replication. So whenever DNA replication is taking place, okay, suppose this is an active phase of replication. Over here there's a replication fork, you can see. So right now over here a strand is synthesized and lagging strand is also being synthesized. At this point of time, if you have a base analog over here, this analog may resemble a normal base. Okay, it may resemble some normal base. Actually this was supposed to come over here and get attached, but by mistake this chemical, okay, it structurally resembles this. Although I made it different, but I want to make you understand that this is structurally resembling it. Okay, let me be more specific. I'll show you with the different color. Okay, let me make like this. Okay, this will be more proper. So can't you see that they are structurally similar? But this was supposed to get attached. Consequently, because of the structural resemblance, what got attached? This got attached. You'll say now, what has happened? Mutation has happened because the wrong base was attached. And what do you call such substances which resemble the bases? You call these substances what? Base analogues. So let me give you one example of a base analog. Example of base analog is 5. I have given you earlier also. 5-bromouracil. 5-bromouracil is a versatile base analog. It resembles a base. Its company name is 5-fluorouracil. Both of these, chemicals, both of these compounds rather, they resemble thiamine. What do they resemble? Structurally, they have resemblance with thiamine. So whenever polymerization is taking place, DNA polymerase may think like this is thiamine, but it's not actually thiamine. Consequently, they will be incorporated into DNA, as I made you understand over here. So it was supposed to be thiamine, but who joined the DNA? Who got attached with the DNA? 5-bromouracil or 5-fluorouracil. Now it's common sense. Normal thing is not over there. The pairing property will be affected. Similarly, let me give you another example. You have 2-aminopurine is there. Okay, this is also a chemical compound. This resembles adenine. This resembles, let me write down so that you will get to note it down. This chemical resembles adenine. Okay. So my dear students, these two chemicals resemble thiamine. This chemical resemble adenine. Can't we say that these two which are in the block 1, 2, 3, these chemicals, can't you call them base analogues? Because they resemble the normal bases. They resemble thiamine and this resembles adenine. Okay. Once, my dear students, these things will get incorporated into the DNA. Consequently, what will happen, my dear students? What will be affected? The, I'll write down base pairing in DNA will be affected. You have a wrong base. That will be affected. Base pairing in DNA will be affected. Consequently, during DNA replication, there are chances of substitution. There are chances of substitution. There are chances of substitution of wrong base. That's what we call what? That's what we call mutation. That's what we call mutation. So, what is in a nutshell we are supposed to remember is base analogues are those chemicals which resemble bases like this. It resembles the base, but it's not the actual base. They resemble the bases and they get incorporated into the DNA during replication. Some of the base analogues are like this, and this is an analog of thiamine. This is an analog of adenine. Once they get incorporated into the DNA, consequently, see, look, they are not the normal bases. Once they will get incorporated into the DNA, that will affect the pairing properties of the DNA. That will definitely affect the replication, and during replication against these bases, DNA polymerase will definitely select some wrong base. Consequently, the newly synthesized DNA will have mutation. Okay. So this was about what? This was about base analogues. Okay. So base analogues, you got this idea. What are we left with? Please help me. I actually forgot what is the last thing. Tell me. How can you tell me this is not a live class? I'm thinking of live classes also. Okay. I have not kept my phone on flight mode, so I hope that nothing wrong will happen, inshallah. So base analog, I made you understand this thing. I hope you have copied that. And the example for base analog, can you give me any? 5-bromouracil. Okay. The fourth one, let me take the dye, acridines. Let me take the acridines, the dyes. Okay. Now, my dear students, what is the speciality with acridines is that, my dear students, number one thing which you need to remember about them. Let me use green. Acridines are basically aromatic. That is basically same thing. Aromatic. They are cyclic compounds. My dear students, you might be wondering that where have you come? I was talking about acridines and where is this all information come on the board. I was actually speaking and I completed my topic. After 15 minutes when I finished it, I then went on to my camera. I went to my mobile phone. Actually, I'm picturizing on my phone. I went to my phone and I was supposed to take it out from the stand and I was supposed to go for the editing purposes. But I saw that storage has become enough and the video has stopped at some point of time. So I have to go back to that place now and I have to explain it to you people again. This happens oftenly with me. This is not a new story. So I have to start this journey back from acridines. Oh my god, I have to go back to acridine, then to hydroxylating agents, then to everything. This happens. That's why it's very difficult for me. It's no worries at all. Anything for you people. Otherwise, I have explained it. But that has not been recorded anywhere. It's okay. Okay, I'm loving it also. So acridines are what, my dear students? I told you that. What are they basically? They are aromatic compounds. Okay, they are aromatic, cyclic, or simply like them, they are aromatic compounds. So when I mean aromatic compounds, how do they look like, my dear students? They'll be having definitely cyclic rings. I'm now repeating the same thing second time. Okay, so they have cyclic rings like these. I'm trying to make it simple. They are not as simple as I'm showing. So these compounds, acridines, they resemble like this. They are like this. Okay, they have got rings and rings and rings and rings. Okay, now my dear students, look what do they do. I'm feeling deja vu because I explained it. So this is the double-stranded DNA. Okay, this is the double-stranded DNA. You have nitrogen bases, nitrogen bases, nitrogen bases, nitrogen bases, nitrogen bases, nitrogen. There, complementary base pairing is there, and you have hydrogen bonds in between. So I'm not showing hydrogen bonds because I want you people to understand that what do these aromatic compounds do? What do these acridines do? They come and they stack in between the nitrogenous bases. So this molecule, this compound will come over here, and it will get stacked over here. See what has happened? This molecule comes and it takes place between the nitrogenous bases. We call it what? It gets stacks. The acridines, the aromatic compounds, the planar aromatic compounds, they stacks between what? They stack between nitrogenous bases. Once they stack between the nitrogenous bases, my dear students, think they are going to remain permanently over there. Inshallah, I'll be teaching you about DNA fingerprinting ahead. I hope this is getting short. So when I'll teach you about DNA fingerprinting, at that time I'll make you understand about a compound called ethidium bromide. Okay, that also behaves in the same manner. Ethidium bromide also gets stacked between the nitrogenous bases, but in that technique of DNA fingerprinting, we use ethidium bromide for the detection of the strands or the fragments of the DNA. So you will come and see this phenomenon taking place in DNA fingerprinting also. This stacking of a aromatic planar compound between the nitrogenous bases, in chemistry it's called intercalation. Interchelation, some call it interchelation. It's called intercalation, intercalating agents they are called. Now think, if something is over here, if something has come between the strands of the DNA, can't you think that these bases now become susceptible to changes? Now there are more chances that this base will get substituted. It will undergo substitution type of mutation because it is now not performing hydrogen bond with other, so it is prone to any sort of change. Maybe this phosphodiester bond will now break and you will have a new base added. Okay, you can see what happening. You can see sometimes there's a chance the base will get deleted. You can see deletion type of mutation happening. Who is causing? What is the causative agent for these three changes? Is the incorporation or the stacking of the aromatic compound. Which is this aromatic compound we are talking about? They are the acridines. So they make the DNA prone to substitution, addition, deletion. All of these are mutations. They are mutations. Or you can look into this in this manner, that once you have the planar compound in between the nitrogenous bases, can't we say that the pairing has been affected? So what has happened over here? Base pairing is what? Is affected. Whenever base pairing is affected, my dear students, there are chances that now mutations will happen. This is the simpler view, okay? So mutations will happen. Maybe if from this DNA you want to get a new DNA, so that DNA is prone to mutations. This was about what? This was about acridines. So the simplest logic is that these acridines, they get intercalated between the nitrogenous bases, and after intercalation they make the DNA susceptible to substitution, addition, deletion type of mutations. Now I will give you the example of acridines, okay? Earlier I explained in a different manner, now I have explained in a different manner, okay? So I hope you got this point. And I hope that it's being shoot. I'll have a look at the camera. Yeah, it's running. So now acridines, example I will quote over here. One of the example is acridine orange. They are basically dyes, huh? Acridine orange. Another is proflavin. Both of them behave in the same manner. They intercalate between the nitrogenous bases. Okay. Fifth type is, although I was supposed to omit this, but I will give you this also. Fifth type is hydroxylating agents. Fifth type is hydroxylating agents. As the name indicates, what they will be doing? They will be causing what? They are the chemicals which will be causing hydroxylation of nitrogenous bases. Hydroxylation is what? It is addition of which group? Addition of OH group. So hydroxylation is done by, examples hydroxylamine, I'll quote that later. So suppose, let us see how hydroxylation happens and how it affects DNA. Suppose I take cytosine over here. You know cytosine? Okay. They are your friends if you are medical students. Cytosine, if you treat it with hydroxylating agent is hydroxylamine. Okay, this chemical causes hydroxylation, and you will get what? The product will be hydroxylamine. Product will be hydroxylamine. Now you say, ma'am, what will happen? Nothing will happen. It's not hydroxylamine, it's hydroxycytosine. Okay, so let me remove this also. This is hydroxycytosine. Okay, hydroxylamine. This is the impact of the temperature basically. I have to turn off my fan and it becomes so hot over here. So this is cytosine. It normally base pairs with guanine. Now see, hydroxycytosine will not base pair with guanine. It will pair with, in place of guanine, it will pair with adenine. What is it? What has happened? Substitution. Okay, if you are smart enough, you will see, ma'am, yes, this is disaster. If this was over here, it would have bound with guanine. This will not do the correct base pairing. It will perform base pairing with adenine, means the base pairing is affected. Okay, if base pairing is affected, definitely there will be substitution type of mutation. So this was about hydroxylating agents. I need to see whether this is, yeah, it's being recorded. So hydroxylating agents cause hydroxylation. As you saw, hydroxylation happening in cytosine, it getting converted into hydroxylcytosine. Thereby, it is affecting the base pairing also. So these five are what? These five are your chemical mutagens. They are your chemical mutagens. So let me quote the example for hydroxylating agents. I was thinking that I will omit it, but I discussed this also. So example is hydroxylamine. Okay, so this was all about your hydroxylating agents. And even my dear students, when we talk about acridines, okay, remember one thing. So I missed this important point. Whenever you have a double-stranded DNA, and what got intercalated, my dear students, I missed this important point. So what got intercalated over here was acridine. Okay, so after this I told you that what could happen. What could happen here? Addition could happen, deletion could happen, substitution could happen. Okay, you know that addition and deletion, whenever these things happen to the DNA, they cause which type of mutation? They cause frame shift mutation. They change the entire frame, entire reading frame. I have made you understand this in my lectures. So frame shift mutation will be caused, or what you call gibberish mutations. Okay, they are very disaster, gibberish mutations. So dyes are leading finally to the frame shift mutation, where there will be disastrous effect on the phenotype of an individual. This point I missed. So this was about acridines, okay, because I have shown you intercalation will cause this, and this will result in a frame shift mutation or gibberish mutation. This was all about your mutagens. So today you understood about what? Today you understood about, number one, you understood was this what basically is spontaneous mutation and induced mutation. What are the chemicals which will induce mutation? They are called mutagens or mutagenic agents. Now then I gave you this explanation for the types of the mutagens. Either they could be physical, they could be chemical. Physical, under physical category we discussed radiations and we discussed what? Temperature. Under radiations we discussed ionizing as well as non-ionizing radiations, whereby I taught you about thymine dimers. Then under this temperature, I told you simply that you have, with every 10 degree rise in temperature, you have 5 times rate of mutation getting increased, okay. Then I have given you this explanation for chemical mutagens, whereby I gave you alkylating agents, deaminating agents, base analogues, acridines, and I gave you information about whom? I gave you information about hydroxylating agents. I have to keep on checking this also. Example for hydroxylating agent is EMS. Example for a deaminating agent is nitrousacid. This example is 5-bromouracil. Example for acridine is acridine orange. Example for hydroxylating agent is hydroxylamine. This was all for your mutations. Now we will consider that mutation we have done. Now my next topic will be, inshallah, I'll be taking up your important question, that is genetic disorders, okay. Genetic disorders I'll be teaching. This is in your syllabus, very important topic. You have to discuss in detail about the Mendelian single gene disorders, and you have to discuss about the chromosomal disorders. Under Mendelian single gene disorders we have to discuss each and every category. We have to discuss autosomal dominant, we have to discuss autosomal recessive, then we have to discuss sex-linked dominant, then we have to discuss sex-linked recessive under Mendelian single gene disorders, okay. Then under chromosomal disorders I will teach you about autosomal disorders, and I will teach you about sex-linked disorders, be it Down syndrome, Calvin-Filter syndrome, and Turner syndrome. I will be teaching you, inshallah, everything now, because you have the whole. It will not take you so much time to understand these things. I'll be giving you, inshallah, details about these disorders. Till then, just explore this information. Don't forget to, like, share and subscribe to my channel. That's very important. And I just want to thank my students, my colleagues, my supporters and encouragers who are encouraging me. Whatever I'm doing is basically for the benefit of the student community. So just try and forward this video to others also, so that all of you will be benefited. May all of you be blessed, and may you be happy in your lives. Remember, stay safe, stay home. So just remember me in your prayers. And that's it all for the day. Take care. Fi aman.